গুড মর্নিং বিশাল,
গুড মর্নিং।
কেমন চলছে? আজকে বর্ষণজিৎ স্যারে কখন গো? অ্যাকচুয়ালি সিগন্যালটা জানি। আচ্ছা আচ্ছা,
একটা মিনিট। আজকে 9:30 থেকে।
9:30 থেকে। আচ্ছা আচ্ছা, আমি তো দেখলাম যে আগে থেকে করে দিলাম। সাগর স্যার কালকে ফোন করেছিলেন, তো বললেন যে কখন হচ্ছে। আমি বললাম আপনাকে আমি লিঙ্কটা পাঠাচ্ছি, ওই ইউটিউব লিঙ্কটা শেয়ার করলাম স্যারের সাথে। তো বললাম যে আপনি 9:30 থেকে আপনি দেখতে পাবেন। তো ঠিক আছে, আমি
মানে কি যতীনদা?
না না না, সাগর সান্যাল স্যার।
ও আচ্ছা 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 আচ্ছা, স্যার।
হ্যাঁ, তো উনি খবর পেয়েছেন কালকে। অ্যাকচুয়ালি উনি থাকতে পারেননি, তো বললো আমি কালকে থাকতে চেয়েছিলাম কিন্তু পারিনি, তো তোমার কাছে কি লিঙ্কটা আছে? আমি বললাম হ্যাঁ আছে স্যার, আমি আপনাকে শেয়ার করবো।
আর তারপরে
আর কি
তারপরে তোমাদের ক্লাসটা কিরকম চলছে?
ক্লাস চলছে, যতটা পারা যায় পড়ে যাচ্ছি। এবারে কবে যে কি কি ঠিক হবে কিছু। আচ্ছা, সেপ্টেম্বর থেকে কি খুলবে কিছু বলছে?
বলা খুব মানে বোঝা ডিফিকাল্ট, কারণ মমতা ব্যানার্জি তো বলেছিল স্কুল কলেজ খুলে দিতে, কিন্তু এরকম তো অনেকবারই বলেছেন, কিন্তু আলটিমেটলি তো লকডাউনটা এক্সটেন্ড করেছে।
হ্যাঁ হ্যাঁ হ্যাঁ,
এবার কালকে থেকে কল্যাণীতে 7 days এর জন্য টানা লকডাউন।
বাবা। হ্যাঁ, যেভাবে এখন ইনক্রিজ করছে নাম্বারটা, সত্যি মানে
এখন
খুব
মানে
ডিফিকাল্ট।
এই গিয়ে মনে হচ্ছে এক্সপোনেনশিয়াল ফেজটা উঠছে আর কি।
হ্যাঁ হ্যাঁ,
এরপর কিন্তু কিছুই হয়নি, এখনই হচ্ছে সবচেয়ে রিস্কি এরিয়া।
সেটাই। দেখা যায় ক্লাস যবে হবে হবে। এখন 1 বছর যদি একাডেমিক ক্যালেন্ডার থেকে পিছিয়েও যায় অসুবিধা নেই, কিন্তু আগে তো প্রাণটা বাঁচতে হবে।
সেটাই, কারণ মানে এখন একাডেমিক মানে কোনো কিছুরই এখন ঠিক নেই।
সেটাই
আগে তো
আগে লোকের জীবন, তারপরে স্টুডেন্টরাও তো মানে এখন কতটা যে আমরা তো পড়িয়ে যাচ্ছি, কিন্তু কতটা যে তারা পড়ছে সেটা তো একটা দেখার ব্যাপার।
হ্যাঁ হ্যাঁ হ্যাঁ।
আর কি বাড়িতে সবাই ঠিকঠাক আছে তো ওখানে?
আপাতত তো ঠিক আছে।
আমার তো বহুদিন মানে অনেকদিন বাড়িতে যাওয়া হয়নি।
আচ্ছা কৃষ্ণেন্দু, এখানে আমার পাশে ওই ইউটিউবের ওই চেকিংটা হচ্ছে, একটু ইকো হয়ে
ও
যাচ্ছে।
আচ্ছা আচ্ছা, ঠিক আছে ঠিক আছে।
আমি একটু অফ করলাম আমার ঠিক আছে।
যারা কথা বলছে দেখো কথা বলছে ওখানে কনভারসেশন চলছে অনেক কিছু আছে অনলাইনে তাদের ও মাইক তো বলো মাইক বন্ধ আছে তোমার সাউন্ড বন্ধ করো এই যে এখানটায় সাউন্ডটা বন্ধ করো সেটা হচ্ছে করে দেবে মাইক খোলা আছে নাকি সব বন্ধ আছে জব হচ্ছে না কোনটা অফ করলে এখন ওখানে কি তোমার ওখানে নিতে হচ্ছে কি মানে জয়েন করো। গুড স্যার, বিশাল বলছি। হ্যাঁ, আপনি হ্যাঁ, হ্যাঁ, হ্যাঁ। আচ্ছা। হুম। নাও। ম্যাডাম অ্যাকসেপ্ট করলাম। তুমি জানি ম্যাডামকে হ্যাঁ, রিচার্জ জয়েন করাই তো আছেন। হ্যাঁ, বলো। ম্যাডাম অনেক কিছু বলিনি। তুমি তুমি একটু বলে দাও তুমি সাজেস্ট করো, আমি একটু বলতে আমি দেখছি স্যার ম্যাডাম দিদিনি তুমি ম্যাডাম আপনি কি জয়েন করতে পেরেছেন? আপনি কিন্তু দেখিয়েছিলেন। হ্যাঁ হ্যাঁ আচ্ছা ওকে। তুমি বলো তুমি ডিপার্টমেন্ট আমার তোমরা কেন করা
বসুন্ধরা কেমন আছো? বসুন্ধরা শঙ্কর বলছি।
Professor Mojumdar, can you hear us? Can you hear us, Professor Mojumdar?
হ্যালো,
Professor Mojumdar, আপনি শুনতে পাচ্ছেন আমাদের? Hello?
Hello, can you hear me?
Yeah, we can hear you, but can you hear us? Your sound is fine.
Hello?
Hello? Yeah, we can hear you. We can hear you.
চ্যাটে লেখ।
হ্যাঁ স্যার লিখছি স্যার। হ্যাঁ
write to the chat.
স্যার কথা শুনতে পাচ্ছেন না।
Hello?
Yeah, we can hear you. We can hear you.
রঞ্জন কানেক্ট করা যাচ্ছে?
স্যার উনি শুনতে পাচ্ছেন না স্যার, আমরা ওনাকে শুনতে পাচ্ছি, উনি পাচ্ছেন না আমাদের শুনতে। একটু রিকানেক্ট করে দেখুক, মানে রিকানেক্ট এখনো হবে, মাত্র 32 পিপল জয়েন, তো
হ্যাঁ
রিকানেক্ট করতে
সে-
পারবে।
হ্যাঁ ঠিক আছে, একটু দেখো বলো একটু।
অফ হয়ে গেছেন, এখন হয়তো আবার কানেক্ট করবেন। বিশাল আছো আশপাশে?
হ্যাঁ আছি আমি।
রিকানেক্ট করবেন উনি।
হ্যাঁ ওকে।
Good morning, sir. Good morning, sir.
মর্নিং, ভেরি গুড মর্নিং।
So now what is the status? Professor Mojumdar, can you hear us?
Hello, can you hear me?
Yeah, we can hear you. You are clear and loud. But
Yeah.
can you hear us?
বসন্তদা শুনতে পাচ্ছ? হ্যালো,
স্যার কি করব? উনি টক দিতে পারবেন কিন্তু কোনো কোশ্চেন নিতে পারবেন না।
দাও একটু ওয়েট করো, দেখো ওর আই থিঙ্ক দেয়ার আর, তাহলে সেটাই বলো।
হ্যাঁ, টকটা শুরু করতেই পারেন, কারণ ওনার ভয়েস তো সবাই শুনতে পাচ্ছেন।
I can't hear you.
রাইট স্যার?
হ্যাঁ সেটাই করো। উনি কি শুনতে পাচ্ছেন আমাদের কথা?
না স্যার, আমরা ওদেরকে শুনতে পাচ্ছি
দীপাঞ্জন?
না স্যার, উনি আমাদের শুনতে পাচ্ছেন না।
তাহলে ওনাকে একটু বলো যে এরকম আমরা শুনতে পাচ্ছি, প্রেজেন্টেশনটা শুরু করা যেতে পারে, কিন্তু আমরা বের করতে পারছি না, কিন্তু টেকনিক্যালি কোনো প্রবলেম হচ্ছে, কিন্তু এটা হবে পরে, এর মধ্যে যদি হয়ে যায়।
উনি লিখছেন যে
হওয়ার কথা না তো।
উনি অডিয়েন্সকেও শুনতে চান, কারণ যারা কোশ্চেন করবেন তারা ডাইরেক্টলি তাহলে ইন্টারঅ্যাক্ট করতে পারবেন আর কি।
সেটা তো ঠিকই,
তো সেক্ষেত্রে অডিয়েন্সরাও কেউ শুনতে পাবে।
একটু কি ওনাকে বলবে কি ইয়ারফোনটা, স্যার আপনি কি একটু ইয়ারফোনটা লাগাতে পারবেন? কারণ অনেক সময় ল্যাপটপে প্রবলেম হয় স্পিকারে বা
উনি
কম্পিউটারে।
শুনতে পাচ্ছেন না, উনি শুনতে পাচ্ছেন না, এখান থেকে বলে লাভ নেই, টাইপ কর।
আচ্ছা
হ্যাঁ
Okay, so in that case, what to do? স্যার কি করব?
না ওনাকে একটু যদি বলা হয় যে প্রেজেন্টেশনটা দিক, ইন্টারাকশন যদি পরে সম্ভব হয় করা যেতে পারে, কিন্তু প্রেজেন্ট তো স্টার্ট করে দেওয়া উচিত।
হ্যাঁ, উনি সেটাই জিজ্ঞেস করছেন। তাহলে
রিকোয়েস্ট করো একটু প্রেজেন্টেশনটা স্টার্ট করে দেওয়ার জন্য।
ওকে, তাহলে লেট মি ইন্ট্রোডিউস হিম ফার্স্ট মেবি টু দ্য অডিয়েন্স।
হ্যাঁ, ইন্ট্রোডিউসটা পরেও করতে পারো, লাস্ট টাইমে তুমি একটু স্টার্ট করে দিতে পারো।
ওকে স্যার।
দু লাইন
আগে
বলে
ওনাকে স্টার্ট করতে বলো। হ্যাঁ
হ্যাঁ।
হ্যাঁ, যে বলো। হ্যাঁ,
হ্যাঁ,
তা নাহলে উনি তো শুনতে পাচ্ছেন না, তাই বুঝতে পারবেন।
Exactly,
হ্যাঁ। Let him start with this presentation. Okay.
বিশাল মাইকটা অফ করো, ফিডব্যাক দিচ্ছি।
Can you see the screen?
যে হ্যাঁ, আমরা দেখতে পাচ্ছি, নাহলে তুমি বুঝতে পারছি না।
Okay. I'm going to start
Yes.
now.
It's coming.
So today I'm presenting hello. So I'm presenting today on SARS-CoV-2, the molecular insights to the potential therapies and vaccines. So why COVID-19 is a pandemic? So this is the chart that shows on March 1st that the average death, like per day, how many people are dying. This is the average of last 5 years on March 1st, that most of the people are dying in tuberculosis. That is 3,000 people per day. And on March 28th, you look, on March 1st, COVID-19 death is 56. And on March 1st, the tuberculosis death is 3,000. On March 28th, the COVID-19 death is 3,200. So, and tuberculosis death is 3,000. So this shows that in rapidly, how many people per day are dying that on COVID-19. So this is the reason why it is called pandemic. So this is today's data that on global death percentage is 3.75%. And in India, the death rate is 2.07%, according to the Johns Hopkins site. So now let's talk about the coronavirus family of virus, that SARS-CoV-2 that that is causing the current pandemic. It belongs to the beta coronavirus group. So the beta coronavirus, this is infecting the humans, but the alpha coronavirus infecting the pig, and also there is gamma coronavirus that is infecting the birds, or the avian coronavirus. So this is the phylogenetic relationship of the SARS-CoV-2 from human, pangolins, and bats. So this SARS-CoV-2 in human is just between bat and pangolin. It is more towards pangolin, but still the genome of SARS-CoV-2 is slightly different than bat and pangolin. And there is a considerable overlap also in the phylogenetic tree. So detection, so this virus is being detected by real-time PCR. That is a very known process now, that you collect the sample from the nasopharyngeal swab, and then you put into the stabilizing solutions, and then you lyse and inactivate and isolate the RNA, which is a bead-based isolation process, and you purify the RNA and then amplified and detect the RNA. So the RT-PCR, now you detect the RNA by RT-PCR. So you reverse transcribe the RNA, make the cDNA, and then do PCR amplification using TACMAN probe, which is a fluorescent probe, and then you measure the quantified the photon from the amplified nucleic acid. So this is, if now you compare directly the delta CT value. If that the delta CT value higher, means that the viral load is less, but if that means the number of cycle, if number of cycle is low to reach the saturation, that means the viral load is high. So that's how we measure. So this is the color contrasted electron microscopic image of the human lung cells that is infected with SARS-CoV-2. So this is a contrasted image, and compared to other virus size, the coronavirus particle is actually, each particle is big, about 100 nanometer. So this is the genome organization of SARS-CoV-2. This is the SARS-CoV-2 genome. The ORF1AB protein is being encoded by ORF1AB gene. This protein first translated as a huge polyprotein, and then getting cleaved by a viral protease, and then called 3CL-Pro, and then a lot of non-structural protein, small pieces being made. So the full-length protein, non-cleaved protein, is not active, but the activity of the protein only comes up upon cleavage. So cleavage is essential. And one of the non-structural protein, NSP12, is actually the RNA-dependent RNA polymerase. There is spike protein encoded by the spike gene. It is the largest glycoprotein, and it forms the spike structure, which is the largest structure also of the virus, and essential for entry into the host cell. So, and also this is the target antigen for most vaccines currently. There are another structural protein. All these proteins are non-structural protein, means these proteins are only present in the infected cells, but not in the mature virions. But spike protein, E, M, and N protein, these are structural protein. These proteins are being packaged into the mature virion. And so the function of E protein is, this is the envelope protein. This functions as the ion channel and extremely required for pathogenesis. There are membrane matrix protein also, that most abundant protein in viral structure. And there is nucleocapsid protein, or N protein, that binds and wraps around the RNA genome to protect the genomic material. Can you listen? Can you hear me?
Yeah.
Okay, so this is a little bit busy slide, shows the whole life cycle of this virus. So when CoV-2 virus enters the human cell by opening the receptor called ACE2, which is the angiotensin converting enzyme receptor, and this receptor is not enough. There are another protease called TMPRSS2. That function of this protease is also required to allow the entry of this virus. Once these are functional, the virus enters the cell, and then it releases the genomic material, which is the positive strand RNA. So immediately, the first job the virus is doing is translating the RNA genome. And upon translation, the first protein is being made is the ORF1AB. the huge polyprotein. And the polyprotein is being cleaved by the viral protease PL2-PRO and 3CL-PRO. That acts like a scissor. And this is extremely important step, because without the activity of the scissor, this polyprotein cannot be clipped. And if it cannot be clipped, non-structural protein, many important non-structural protein cannot be made and virus cannot survive. So this is a very important step in viral life cycle. Once these non-structural protein is being made, one of the protein I told that it is NSP12, which is the RNA-dependent RNA polymerase. The first job of this protein is to reverse transcribe make the cDNA copy of the genomic material. And then once that part has been done, so then again the replication complex acts on it, and again prepare the genomic material, which is again now the positive strand, and now it will make the segmented mRNA. That all these mRNA are encoded viral protein. Most of these are structural protein like S, E, M, N. Once this protein is being made, by exploiting the cellular translation machinery called ribosome, and this protein will immediately engage into the ER and Golgi pathway, because these are the membrane protein and their destination is in the membrane. So immediately they will engage into the ER Golgi pathway, and using this pathway, they will arrive at the membrane by systematic processing through the secretory pathway. So what virus is doing? Virus is occupying the protein synthesis machinery and also exploiting the cellular secretory pathway. And then mature virion will be made, and then it will release from the cell. It can be slowly released from the cell, or it can, if the load of the virus is high in the cell, it can burst open the cell. So, and then these newly made virion can infect other cells, and that's how the infection propagates. Now the question is, why this SARS-CoV-2 is highly infectious? So in order to know this, we have to know how this S protein enters the cell. The S protein has a subunit called S1 subunit, which is, if you consider the S2, which is the angiotensin converting enzyme 2, which is the receptor that is a lock, and then the S protein is a key. So it is a very efficient key. It can open up the lock by 1 out of 1. Like the chances are very high that it will open up the lock. So, and why? Because of its high affinity. So the answer is affinity, affinity, and affinity. So now one thing I would like to mention here, that the SARS-CoV and MERS, SARS-CoV, not CoV-2, only CoV, SARS-CoV, the previous SARS-CoV, and MERS was more lethal but less contagious than SARS-CoV-2. So why? The answer you will see in this paper published in Cell on May 14th. What the investigator did is they did some protein interaction assay through Biocore using the CoV-S recombinant protein and S2, and they have just simply measured the KD value. The KD value of SARS-CoV-2S to S2 is way more negative than the KD value of SARS-CoV-S or MERS-S. So since the KD is less, affinity is high. So the relationship between KD and affinity is inversely proportional. So that's the reason why SARS-CoV-2 can enter the cell very efficiently by using the high affinity binding of S and S2. So why this CoV-2 targets multiple organ of human? Because many cells of human organ expressed S2 receptor. So it is almost ubiquitously expressed. Nasal passage cell, lung cell, that secretory basal and multiciliated cells, ileum, our intestinal tract cell, that epithelial enterocytes, all of them, even heart cell, eye, corneal epithelium, like kidney cells, pancreas, islet cells, they are also expressing S2. So all of these organs are really target of this virus. And with consistent to this, now there is lots of data are mounting clues that suggest coronavirus may trigger diabetes because it inhibits islet cell activity, and also organs that participate in glucose metabolism shows high S2 expression. And actually, for this, there is actually a registry called COVID-Diab registry, maintained by King's College of London, where they have a repository of COVID, how COVID is creating diabetes. So they have a repository of large data directly collected from the patients. Now I'll come, so I'll ask you time to time, can you hear me? So there is another interesting aspects of SARS-CoV-2 is you might see in the web or newspaper, there are conspiracy theories are also going on. So is it a man-made virus? So is the highly infectious property of human SARS-CoV-2 is created by deliberate genome engineering? So now I will present some evidence that why it is not. So what you are looking at here is the genomic material of this virus. So this is the spike. Now you slowly concentrate on the spike gene. So you will see the spike gene is divided in S1 subunit and S2 subunit. S1 subunit is the receptor binding domain, and there is a polybasic cleavage site just at the junction between S1 and S2 subunit. And this polybasic cleavage site is being recognized by TMP-RSS that facilitates the infection. So now I am requesting you to look at the receptor binding domain sequence of human SARS-CoV-2, bat, pangolin. So all of these, so sequence of the receptor binding domain of the spike protein S of human SARS-CoV-2, bat SARS-CoV, and pangolin SARS-CoV, it showing remarkable conservation. There are only little difference. So this actually shows that the virus is continuously trying to jump the species barrier by mutating the S protein. So this is a process driven by the natural selection. So I have collected these from the journal Nature Medicine, published in 17th March issue. So, but still, there are some fuel in the conspiracy theory. So now I will focus your attention to what is still why the conspiracy theorist is not satisfied by this. So what they are telling is, look at the polybasic cleavage site, and polybasic cleavage site, all of a sudden you will see these 4 insertion of amino acid. So this insertion is still fueling the conspiracy theory. So why there is extra 4 amino acid insertion at the polybasic cleavage site? So this feature is a new feature, and this is not present in any other species. So hence, so their logic is, hence this is engineered. So that's their strongest logic. Well, I'll show you some data why it is not. So this is more recent data published in May 11th issue of Current Biology, is a Cell publication, that what they have found, a novel bat coronavirus that is closely related to SARS-CoV-2 that contain natural insertion. So I will request you to focus on this. So this is the SARS-CoV-2. These are the insertion. And in bat also this insertion is going on. So showing natural insertion of amino acid in the polybasic cleavage site of the S protein, so which really shows that it is possible that in nature also these multi amino acid insertion can is going on it is possible. So this is the strongest point to debunk the conspiracy theory. So now I will talk a little bit about the structural aspects of the infection. So this is a reconstructed image showing the cryo-EM data, cryo-electron microscopic data from the protein data bank that shows the moment of entry of the SARS-CoV-2 virus with the human cell. This is the receptor protein ACE2, so reconstructed from the PDB data file. And also, this is the spike protein of SARS-CoV-2. This is also reconstructed from the PDB protein data bank data file. So now I would like to mention that the structural biology is bringing actually a real revolution in the discovery of future therapeutics in an unprecedented speed. So because the structure of spike protein and the ACE2 has been solved up to the 3 angstrom atomic resolution level by cryo-electron microscope. So what you are looking at, the structure of SARS-CoV-2 S protein receptor binding domain, and this is the ACE2. And you will see now using artificial intelligence, scientists now are designing many small molecules that will fit into this space. And those molecules could be the potential drug that can block infection. So many of them are in human clinical trial. I'm showing this from the article published in Science, that 27th March issue. I told you that the Caesar is a very important part of this virus. So what happened if you target the Caesar? So again, here is structural biology played a main role here, that they identified a highly potential drug called alpha-ketoimide that significant promise in human clinical trial. So how this drug works? So this drug actually targets the protease Caesar of the virus 3CL Pro. So this has been first in silico designed, and you can see that the structure can be well fit in the atomic structure of 3CL Pro. So the gray part is the atomic structure of 3CL Pro, and the colored is the alpha-ketoimide. So this has been done first in silico, and then they synthesize the compound, and then they have tested the antiviral efficiency of the compound, and it gives very promising result in preclinical setup, but the human clinical trial result is still pending. So this is a cryo-electron microscopic structure of the RNA-dependent RNA polymerase, the heart of the virus. That here you can see it has a different domain: finger domain, thumb domain, palm domain, beta hairpin, neuron domain, and this is the interface with other non-structural protein. So this is the whole structure of the RNA-dependent RNA polymerase. And this is the RNA-dependent RNA polymerase that bound to the remdesivir. So this is the remdesivir, which is actually a nucleoside analog, and how it inhibits the polymerase, it just incorporates itself. And when it incorporates itself into the chain, and the chain then terminates. So this is how the remdesivir works, and it currently showing very good result, and also it got temporarily approved by FDA for emergency use on critical patients. And there is another structural and analog of remdesivir is the favipiravir. So this is also being used in many countries, especially in Japan, originally used as an anti-flu compound, but now it is also showing high potency against SARS-CoV-2. Now, quickly, I will touch some little bit complex aspect of this SARS-CoV-2 on the future drug discovery aspects. So this, and in order to understand the future drug discovery, I would first like you to know how the virus uses its own protein to break the communication between the cellular protein to protect its best interest. So if you consider a cell, in cell there are thousands of protein in a human cell that is communicating all the time between each other and keeping the cell healthy. So this is a healthy condition. Now, when virus enters the cell, now consider this virus as a colored ball protein. These are the viral protein, and the ash colored ball is the cellular protein here. And now the square shaped is the cross point between communication of viral and cellular protein. So a potential future antiviral, its job will be to break the communication between the viral protein and the cellular protein. And how this is happening? So I would request you to draw your attention for the time being into this paper, which is a very recent paper published in 16th July issue in Nature, that is SARS-CoV-2 protein interaction map. The genome-wide interaction map of human protein and SARS-CoV-2 has been solved, and it reveals many targets for drug repurposing. What is repurposing? Means whether it is possible to treat this disease by using the already existing drug for other disease. So what's the advantage? Because if it can be tackled by already existing drug, that means by discovering the repurposed drug, you don't need to carry a long clinical trial because this drug is already approved. You know, this is the most advantage of this. So I would suggest you to draw your I'm requesting you to see this, but I'm not telling you to look at all the protein. It is impossible, you know, to look at all the protein. So what I'm telling you is the red square is the viral protein, and these small spots are the cellular protein. So these interaction map shows every viral protein what cellular protein it interacts. And now, and not only that, this interaction map also shows what are the potential drugs that can interact with the cellular protein that is in turn interacting with the viral protein. That means what could be the promising drug that can intercept the communication between the viral protein and the cellular protein. And actually they have found a many group of drugs that can function this way. So with that, you know, sorry, this is again a little bit complicated slide. So and they feed the data in the machine-driven artificial intelligence that has been used to discover anti-SARS-CoV-2 activity. So from the repurposed drug. So why it is repurposed drug, I'm telling? That means the drug is already in use for other purpose. The one of the advantage of using those, of looking at those drugs, because in the GEO site, in the gene expression omnibus site, it is already a vast amount of data has already been deposited. That what is the genome-wide gene expression changes in response to each drug. So what the artificial intelligence is retrieving those data and then connecting the balls. That means, and then predicting that which drug can intercept the communication between the colored ball and the ash colored ball. That means the viral protein and the cellular protein. And a complex network between the ash colored ball and colored balls, these are being analyzed by artificial intelligence, and they are predicting a cohort of drugs, already existing drugs that being used for other diseases that can have potential anti-CoV-2 activity. So which is really bringing a revolution in the drug discovery field. Again, I will ask, am I visible? Can you hear me? Hello, can you hear me?
It's all fine. It's all fine.
Okay, now I will switch to the immunological aspects of this talk. that how the immune regulation of this virus can proceed. You know, because there is prior knowledge of the immune regulation of SARS-CoV and MERS. This is enormous help to predict the immune response by SARS-CoV-2. This is a generalized figure, and what I'm showing here, one part is the innate immune response, and this side is the adaptive immune response. So in innate immune response, when SARS-CoV-2 enters the cell, so it opens up like a spectrum of immune response pathway. So there are 1, 2, 3, 4. So 4 pathway got triggered. So first pathway got triggered by the ion channel proteins that activates NLRP3, caspase 1 that forms pro IL-1 beta to IL-1 beta. This is and causes inflammation. So also virus, when this virus enters the cell, it opens up the single-stranded RNA. That single-stranded RNA activates MYD88, NF-kappa B dependent pro-inflammatory cytokine pathway that produces IL-6 or TNF-alpha. And not only single-stranded RNA also form double-stranded RNA. And when double-stranded RNA formed, it triggers RIG-I, MDA5, MAVS mediated IRF7, IRF3 dependent transcription that ended up in producing interferons. And not only that, there are JAK-STAT pathway that is also causing production of RNSL and many other pro-inflammatory cytokines. So this is happening even without producing the antibody. Immediately when virus infects the cell, as a defense, cell is making a plethora of inflammatory cytokines as a defense. But in the adaptive immune response, when it switches from innate to adaptive immune response, so what happens? The virus being recognized by the first cell of the adaptive immune response is the macrophage. So macrophage, which is the antigen presenting cell, it grabs the virus, it digests the virus, and then display the viral antigen in the surface of the macrophage or the antigen presenting cell. That attracts naive T cell. So naive T cell got triggered by the presented viral antigen and formed CD4 plus T cell and CD8 plus T cell. So CD4 plus T cell now induced B cell by through the humoral response, and B cell forms plasma cell, and plasma cell then making antibody, and this antibody can neutralize the virus. So this is a really the 101 of the innate immune response and adaptive immune response. But there are more to it. So virus, you have to understand that the virus itself do not cause any damage to the human. It is the hyperinflammation that causes the overactive innate immune response. That is causing the cytokine storm, so that it cannot be controlled. And that is the reason why people some of the infected people are going into the ICU. So this is a severe lung pathology figure that I'm presenting here. This is the X-ray of an infected patient lung. So, and this is the post-mortem sample of an infected patient. It is a color contrasted electron microscopic image that you can see that the SARS-CoV-2, how SARS-CoV-2 invade these the lung cells. So lung has like alveolar, so which is alveolar cells, and when the lung got inflamed, high expression of this inflammatory cytokine causes cytokine storm, and this cytokine storm causes the secretion of mostly water from this alveoli, and then it rapidly filled up the bronchoalveolar space with the lavage fluid. So in normal lung, there are rapid carbon dioxide oxygen exchange is going on without any hindrance. But when lung is inflamed by virus, so it filled up with lavage fluid, and then the patient cannot breathe and then needs intubation or ventilation. So this is the reason why people are dying mostly by infecting in this virus in the ICU admitted patients. Now, I will tell you a little bit about what causes this over-inflammation. So in order to understand that, I would like to tell a little bit a mysterious, a mystery behind these inflammatory cells. So there is a large number of immune cells. Immune cells are not homogeneous. There is large number of immune cells that is showing with varying status of inflammation. So it making the system very plastic. And there is a dynamic equilibrium between the inflammatory immune cells and the anti-inflammatory immune cells. Now, the relative numbers of these diverse immune cells determine the resultant inflammation in the infected patient's body. Whether it will be lethal or not, that is driven by how well the equilibrium could be maintained. So in the absence of this homeostasis, that is causing the cytokine storm in the lung. So this is the reason for hyperinflammation and death for majority of the SARS-CoV-2 infected patients. So this, how to maintain this dynamic equilibrium well is called trained immunity. And the people who are staying relatively in germ-free condition, that means the so-called healthy condition, in most of their life may not have trained immunity. So their balance is poorly balanced. Whenever they see any infected organism like this virus, so the inflammatory cells go ballistic. This is the reason why in Western countries, many people are dying in this. Because of mostly staying in the germ-free condition. BCG vaccine, you might have known that BCG vaccine that against tuberculosis, but not only tuberculosis, this vaccine is also is a potent inducer of trained immunity, and this is the reason why BCG vaccine, is giving significant protection against this virus. They recently mostly completed human clinical trial in Australia that found that more than 50% of the patient got protected by this vaccine. And I believe India also a protection, a clinical trial is going on here. And BCG actually is a potent inducer of trained immunity, and there are many examples and documented evidence in favor of that. So for COVID-19, there is a huge difference in the pathological response between the individuals. So why some individuals are getting really extremely sick, and why some individuals are tackling it very well. So the scientific reason was still unknown, but only paper that published in New England Journal of Medicine shedding some light on this on June 17th issue of New England Journal of Medicine, that they have conducted genome-wide association study of severe COVID-19 with respiratory failure. So and these studies has been done, carried out by scientists from Italy, Spain, Germany, and Norway. So what is GWA study? So this method is based on sequencing the whole genome. So it searches the genome for small variation. It's called single nucleotide polymorphism. And using the statistical power and big data, so they build a significant relationship between the disease severity and SNPs. Actually, they have analyzed a huge number of SNPs, 86 times 10 to the power 5 SNPs, by sequencing the genome of 2,000 COVID patients. And now they have identified actually a treasure trove from that analysis. For that severely ill patient, or the patient who died on account of COVID-generated cytokine storm, they have identified specific SNPs of 6 genes in chromosome 3 locus. So I'll show you the data here from the NEGM, that this is the log p-value showing the significance, and this is the chromosome. So you can see in the chromosome 3, they have identified in a particular locus, they have identified 6 genes that is all carrying SNPs. So what could be the potential connection between the SNPs in this cluster of genes and COVID-19 disease severity? Because all of these patients that undergo have very severe disease. Many of them died. Most of the proteins that is encoded by these gene clusters are inflammatory proteins. And the expression of these proteins are supposed to be very tightly regulated by the regulatory sequence. And little SNPs can tweak the sequence and then make the expression of the cytokine ballistic. So this could be a potential reason why there is some strong individual variation and association has been identified from one individual to other individual to the disease severity. So now, in the last part of my talk, I will talk about the vaccine. So, which is against what is vaccine? So, this is nothing but the natural immunity, mimicking the natural immunity by artificial way. So, in natural immunity, so this part is the now let's focus on the adaptive immune response. So in natural, if you want to be immune, so mostly you can get immune by getting infected by the virus, but this has a price to pay. So, if you don't have a trained immunity, you may be seriously ill. So vaccination is a process of inducing artificial immunity by using a structural antigen or a weak version of the virus by exploiting the same adaptive immune system. So let's talk about first the Oxford vaccine from the Jenner Institute. So what they have done in Oxford vaccine, they sequenced the SARS-CoV-2 spike protein and then cloned the spike protein into the adenovirus vector. This adenovirus vector causes the cold symptom in chimpanzee, but do not cause that much disease in human, do not cause any disease. So now they prepared the recombinant adenovirus and injected into the human. So, and then the same pathway will trigger. That means, the virus will infect the cell, and then macrophage will digest the virus and they display the viral antigen and recruit the T helper cell. T helper cell will induce B cell. B cell will produce antibody, and also T helper cell will induce cytotoxic T cell. So that is the expected pathway. And also, this pathway should also produce memory B and T cell that will live long and patrol the body in case of future infection. So that is the expectation of this. And also the cytotoxic T cell. So the way vaccine works, not only based on antibody, but also based on the generation of cell-mediated adaptive immunity. That means producing cytotoxic T cell that can infect the virus, that can kill the virus-infected cell. So this is the mRNA vaccine platform from the Moderna. The Oxford platform is based on adenovirus, and this is the mRNA platform that is being used by Moderna. And what is this mRNA platform? That is the mRNA of S gene being produced and coated by the lipid particle and the lipid nanoparticle, and the nanoparticle and the RNA complex is injected into the human body. And this RNA will go inside the cell because this lipid nanoparticle will easily fuse the cell membrane because of the hydrophobic property, and then eject the viral RNA in the cell. It will be recognized by cellular translation machinery, prepare the viral protein. Once the viral protein being made, and then it will, again, the same adaptive immune response pathway will be activated. That means the macrophage will get the virus, will get the antigen, display the antigen in the surface, activates the naive T cell, CD4 plus T cell, CD8 T cell, cytotoxic T cell will be made. CD4 plus T cell will induce B cell, and B cell will produce antibody. And that's how we get protection. This is one way, and another way is to use the live weakened virus. And that strategy is also going on. using Codagenex USA and Serum Institute of Pune. They are working on this, and this is a weakened form of virus. That means this virus replicates in the human body for some time that never cause disease, but induces strong immune response. So this is a sort of virus. You can get the virus by passaging the virus in human cell for a long time, and isolating virus that causes significant mutation that cannot cause disease, but little bit replicate in the cell and produce the immune response, and that's how you get protected from the real virus. So this is a different step of the clinical trial of the vaccine. So first step is you isolate a viral particle and you have to adapt it for vaccine. How you adapt it for vaccine? You can weaken the virus. That means it is called attenuated virus. It could be live attenuated virus also. That's the polio virus vaccine is a live attenuated virus vaccine. So, or you could kill the virus by formalin or even heat inactivated, or you can isolate different piece of the virus, or you can map the virus genetic sequence and then clone the virus in different vector. It could be DNA plasmid vector, it could be RNA a lipid complex, or it could be adenoviral vector. And then you disperse these into a solution, and then you test this solution in preclinical trial. That means in cell culture media, whether it can stop the infection of this virus, or then you test it into the mice model, whether it is toxic. If it is no, if it is yes, then you cannot move forward. That is stop testing. If it is not toxic, then you move forward, and then you see whether it can produce sufficient antibody. If the answer is yes, then you go to the human clinical trial, phase 1 trial, phase 2 trial, and phase 3 trial. So in phase 1 trial, you will see whether it is producing sufficient immune response and whether the vaccine immediately after injecting it is not giving any contraindication, or whether injecting the vaccine itself is safe. And now you conduct a little bit, this is between typically 10 to 100 people, and then you conduct a little bit more number of people. The same thing, is it safe? Is it causing immune response? And also you fix the dose. What dose is causing optimum immune response? And then you move to if the answer is yes, then you move to the phase 3 trial. And in phase 3 trial, you will test it on multiple people, like vast number of people, more than 10,000. So Moderna's trial is going on now, and both Oxford trial is also going on now, more than 30,000 participants. 30,000 is in US, but also in other countries. I'm sure it is more than million participants total. So now there are several challenges also in the vaccine road. So say if the intensity of the pandemic goes down, that might impair the clinical trial. So without its real application in the field, it will be really hard to assess the efficiency of the memory B and T cells. So, and also another aspect is SARS-CoV-2 is mutating. If it adopts mutation that in the amino acid residue that is relevant epitope for the antibody, then the vaccine cannot recognize it. So that is another potential problem, and this problem is generic for all vaccine. So and also there are some reports that COVID-19 infection also reducing the T cells. So for some people, it may not give a protection. This could be the cause of why some people are getting reinfected. Although the number is very less, but this is an issue that why it is happening. So there is no definite answer, but it is already known that COVID-19 infection for a very small number of people is reducing the T cell, and that could be the reason for that. So there are dark side of fast tracking the vaccine development. You will see now many people are telling, oh, you need fast track the vaccine development. Maybe you don't need that much scale of phase 3, and it may cause a potential dangerous situation. Because we should not and must not forget the dark history of respiratory syncytial virus vaccine development, where the vaccinated children got seriously ill. I repeat, the vaccinated children got seriously ill when they got the real virus, and some even died. Why? So the history of vaccine is bright for pox, polio, mumps, rabies, but it is not so bright for respiratory syncytial virus, dengue, Ebola, HIV. So the main reason for this is the antibody dependent enhancement. So this is a really dark side of the vaccine, that the real and documented episode behind the saga of many vaccines. But ignoring the requirement of a large-scale double-blind placebo-controlled phase 3 trial, will reduce the chance of catching the antibody-dependent enhancement. So this could bring a bad consequence. So that's how we should not stop phase 3. All vaccines should go a stringent phase 3 on a large number of participants. So what is antibody-dependent enhancement? So this is, if the antibody is neutralizing antibody, it will cause the virus, it will bind the virus, then it will terminate the virus. So that's it. So you get the beneficial effect. What happened? There are non-neutralizing antibodies present. So non-neutralizing antibody will also bind the virus, but this binding will not inactivate the virus. What will do, the non-neutralizing antibody in one hand will bind the virus, and in other hand will bind the cellular Fc receptor. And this will cause the bridging of the virus and the cell. So it will recruit more virus to the cell. So this is called antibody-dependent enhancement, which should not happen. And a detailed phase 3 study will catch this. This is why it is important to pursue a detailed phase 3 study for all vaccines. So this is the vaccine trackers that from July 31st, that 140 candidate in preclinical trial, only one is approved in China for military use, but phase 6 is being phase 3 in very close to completion. So 6 vaccine, and these are the potential players in the vaccine market. Recently, phase 1 and phase 2 trial of the Oxford vaccine has been published in Lancet, which shows that it is safe to the volunteers, and also it causes humoral and cellular immune response. So, am I visible? I'll just can you hear me? And also the Moderna vaccine trial, also they have published their preliminary report in New England Journal of Medicine, phase 1 and 2. That also shows that it causes immune response, producing neutralizing antibody, and on a small number of volunteers, and they did not cause any really safety concern. So these are the vaccines in phase 3 trial. Moderna USA, that is the mRNA-based vaccine. BioNTech in Germany, that is also mRNA-based vaccine. AstraZeneca, University of Oxford, UK, adenoviral-based vaccine. CanSan Bio in China, adenoviral-based vaccine, and these two, Sinopharma and Sinovac from China. All are in phase 3 trials. So in India, as you all know, that Bharat Biotech is also proceeding with the Covaxin, which is an inactivated form of coronavirus vaccine, and also Zydus, which is a DNA-based vaccine. So instead of RNA, they are injecting the S protein sequence into the plasmid and injecting the plasmid, so which is a DNA-based virus. These two are in clinical trial in India. And recently, I have noticed some negative publicity on the vaccine spread by some media that vaccine may not be effective against SARS-CoV-2 because of low stability of the antibody present in serum of patients recovered from COVID-19. So I would like to say that these people are missing a major part of the vaccine immunology that relies not only on the stability of the antibody, but also on the cell-mediated adaptive immunity. So this part is still there. And the maintenance of the memory T cell part is still there. So even the antibody stability is less, if still the vaccine could be effective because of the cell-mediated adaptive immunity. And in consistent with that, the Moderna mRNA vaccine published again another result in 31st July in New England Journal of Medicine. This is an extremely encouraging result on, this is actually a challenge study. So they vaccinate the monkey and then they challenge the monkey with SARS-CoV-2 virus, real SARS-CoV-2 virus, and found that vaccine-mediated virus neutralizing antibody, vaccine-mediated activation of CD4 plus type 2 helper T cells. So cell-mediated adaptive immunity is active, and also strong evidence of CD4 plus T cell-mediated B cell activation. So, and not only that, the speed it is going on a very high speed. August 5th, Nature published that the Oxford vaccine monkey studies has been conducted very successfully. Induction of strong humoral and cellular immune response, mostly Th1, not Th2 response. This is good because Th2 response actually contributes to the ADE, which is bad. That means antibody-dependent enhancement. So this is not happening because of the absence of Th2, which is extremely good result. So significantly reduced viral load in the lavage fluid and respiratory tract, and no significant lung pathology. So both, these Oxford vaccine result and mRNA vaccine result of Moderna are good in the monkey trial, and because the monkeys shows rapid protection. And another good thing I would like to tell all of you that, we all have pre-existing immunity to the SARS-CoV-2. Even we are not exposed to SARS-CoV-2. And that's being published in Nature Review Immunology with many strong evidence that if you are infected with common cold virus, you have to remember that among common cold virus, many of them belongs to the coronaviridae family, not SARS-CoV-2, but coronaviridae family. And they have some homology with SARS-CoV-2, some domain, and T cell already being made. So, and this T cell can show protection against real SARS-CoV-2. So, and this is particularly relevant for the country that is staying in less sterile condition. So, these are very good news for a country like India. And also, there are targets of T cell response of SARS-CoV-2 coronavirus in human. This is independent report published in Cell, also recently in June 25th, that even in unexposed individual, and they have also concluded that other virus can also activate CoV-2 specific T cell. So this is, I think this is the reason why there are a lot of asymptomatic patients there. That means not showing any significant pathology because they have already equipped with these cells, and their cell-mediated adaptive immunity is very high. And another thing that I would like to tell that and that whether the asymptomatic patients are really spreading in high rate, because no, I have not seen a single result that asymptomatic patient in nasal swab has been identified and measured for infection competent virus by plaque assay. That, see, all they have done is RT-PCR assay. So RT-PCR will only tell the presence of of a piece of a virus. Whether it is dead or live, it will not tell. So only plaque assay can tell. But without doing the plaque assay, I don't think it is fair to conclude that asymptomatic patients are actually spreading in high rate. I think the patient who are spreading, they're in high rate, that is, they are pre-symptomatic patient. So they will be symptomatic, but before development of symptom, they are moving around and spreading, not the asymptomatic. I think the asymptomatic patients are not allowing the virus to grow very high in their system. But there are no definitive answers, but lot of questions there in that. So this is the last slide. What I'll tell you that I'm a volunteer of Moderna mRNA vaccine trial, and I'm getting the vaccine in August 11th as a part of the phase 3 clinical trial. And I have already signed the informed consent form. And what I'm telling that in India also the Oxford vaccine trial will going on. There is a recent the news that I have heard that Oxford vaccine trial, they have made an agreement with the Serum Institute. So they will prepare under the license of AstraZeneca, the Oxford vaccine in India, and also they will conduct a clinical trial in India. So I will also encourage, if you get a chance, be a volunteer. So the sun of the COVID-free era will rise because science has a destiny of winning under challenges. Thank you.
I would like to thank Professor Mojunder for his kind and nice deliberation. It was really very informative, although I know that he can't hear me. So as we are running late, maybe we will proceed for our next talk with Professor Keshav. Keshav Mukherjee. So he is from Medical College, Jawaharlal Nehru Medical College. Without wasting time, because he is already known to us, I would like to invite him to deliver his talk. Sir, please. Sir, are you here?
Yeah, sorry, sir, it was online. Somehow the net got disconnected. I'm getting for I'm connecting the net again. Somehow the net got disconnected just now. Sir, is there?
Sure,
just
sir.
Give me a minute. Huh?
Thank you, everybody, for your kind attention. It is 1 o'clock in the morning now here in Cleveland, Ohio. So it is really a pleasure to join you. And, I'll be happy to answer any questions. So I know, I can't hear you. So this is too bad for, without getting the feedback from the audience, it is compromising my motivation.
Yeah, exactly, really, sir. But we can't do anything right now because
it's a pleasure to I can see so many of you, and many of you is already known to me. Hello, Shankar. Is Professor Ghosh is here? Hello.
Yeah.
Okay, then, if there is no other question, I don't know how to tell I can't hear. So I'm not, I know this is very hard to communicate in this thing. But I'm glad that at least you guys hear my presentation. So that's at least that is the good part. So I'm reading the comments. Nice of you. Thank you. Thank you. Thank you.
No.
Yes, sir. Please, can you share?
Thank you, everybody, for your time. So, is any other speaker today?
Yes, sir. There.
Okay. Then, good morning over there. Although here also morning, 1:14 a.m. So, thank you.
Here, sir.
Hello.
Yes, sir.
So is it my turn to speak? I'm Professor Mukhopadhyay from College of Medicine, Geneva Hospital. Or you're settled? Good morning, sir. Am I audible?
Yes, you are.
Good morning, sir. Please.
Yes. Yes, good morning, Professor Mukhopadhyay.
Thank you very much, sir. We had a very nice discussion on listening. Very good opportunity to listen from abroad. It's a very good initiative taken. Thank you, sir. And you have given me a chance to speak on the precautions and safety of the measures for the COVID-19. I think must have been taught, discussed in the last few classes, what you have said in day before yesterday night. So let me start. In order to realize, in order to understand the different safety measures, one must know what is epidemiology. In fact, epidemiology is the study of the distribution and determination of the health-related state or event in a specified population, and application of this study for its control. And the different approaches for the epidemiology are, number one, asking questions and making comparisons. What are the different questions you can ask? What is the event? That is the problem. And here the event is the COVID-19 pandemic. And what is the magnitude? As I said, is a pandemic throughout the world is concerned with this COVID-19 pandemic. Where did it happen? It happened throughout the world. No country is spared. When did it happen? It started sometimes in November or 9 December 2019, and is still continuing. And who are they affected? We all are affected. Why did it happen? There are so many questions regarding this. And the second thing one has to do is the making comparison. Actually, these approaches are the managerial approaches in order to prevent the epidemic. Now, what can be done to prevent it? This is the first question. And how to prevent in future? What to do if I get similar episode or similar incident in the future? What to do? Action to be taken by the community. It's a very good big question, because we all ignored what has to be done. We ignored. And what are the resources and activities required for the organization, and what difficulties may arise and how to overcome it? These are the managerial questions. Please allow me to pick out the mask because it's causing problem. I'm alone in my room. And one of my colleagues who is staying, who is almost 15 feet away from me, who has prepared all the slides for me. Now, this is how and disease spreads. First, there is a reservoir, just like a water tank. From there, the water goes to the resource at the site, at the kitchen. In the same way, this is reservoir. And what are the reservoir for this COVID? This is human. Animal reservoir is, whether it is there or not, is doubtful. There is no relevant literature. It is said it was earlier in the bats and others, and non-living. This is also doubtful if non-living resource or reservoir is there or not. Now the human reservoir, it may be the primary case, the first case, or the secondary case. Or it may be carrier. The carrier means one who carries the disease and spreads to the community. It may be incubatory period carrier. Incubatory carrier means that means right from the beginning of introduction of the infection to the manifestation of the disease. This period is called the incubatory period. In this period, there are certain diseases which are carrier, and there are certain diseases which are not carrier. There are convalescent carrier. That means when the patient is getting rid of the disease, on that time they may be carrier, or it may be healthy carrier. In case of COVID-19, all the three varieties of carrier are available in the community. Or it may be clinical, it may be subclinical, or it may be healthy carrier. I have seen one of the drivers who was totally fit. He has gone to test for the RT-PCR and he was found to be positive. In fact, a healthy carrier doesn't have any signs and symptoms. So the healthy carriers are also there. So one should be aware. Therefore, precaution is very important for the spreading of this disease. Now, regarding transmission, there is no concrete documentation how it is transmitted except the direct contact, or there are some literature which says it's spread by the droplet. Contact with soil, there is no documentation. Inoculation of the skin, it is somebody says yes, somebody says no, and placental this transmission is not well documented. It may be indirect transmission like the vehicle means waterborne, or the vector-borne like the mosquito spreads the dengue, there is no such evidence. Airborne infection, there are some literatures coming out. Fomite-borne, that means this bedsheet and others, whether it spreads it not, is not exactly known, but one should take the precautions. And unclean hands, this is definitely one of the cause of concern, so one should wash the hand regularly in a regular interval. Now, when we talk about the epidemiology, it is always taught in three. It's called the epidemiological tracks. First is the agent, second is the host, and third is the environment. Now here agent is the COVID-19 virus, host is the man because they are suffering a lot, and the environment, whether the environment suits the spreading of the disease or not, this is also very important. Now, the COVID virus is a large single-stranded RNA virus with clown-like appearance due to presence of spike glycoprotein on the envelope. Its orders is also coronavirinae, family is coronaviridae, subfamily is nidovirals, genus is alpha, beta, delta, and gamma, and it causes respiratory, enteric, hepatic, and neurological manifestations. Now, what are the common signs and symptoms of the coronavirus? The common signs and symptoms of the fever, there may be dry cough, there may be tiredness, and there are some uncommon signs and symptoms like aches and pains, sore throat, diarrhea, conjunctivitis, headache, loss of taste, skin rash, and its incubation period is 5 to 6 days, maximum is 14 days. These all has got some applied importance because of this, how many days of quarantine is required or lockdown is required. If you don't know what is the incubation period, you will not be able to set strategy for this. This is a lockdown period, because if you can set a lockdown period, the maximum incubation period, I hope there's a chance to inhibit its spread in the community. This is my personal view. Now the prevention. So what are the different prevention methods? What one can do? Homestay, what is lockdown? Then second is the wearing mask. Third is social distancing. Third is repeated hand washing, hand alcohol rub, no touching of eyes, nose, mouth with non-sanitized hands, and surface disinfections like ether 75%, alcohol 70%, chlorine containing disinfectant, peroxyacetic acid and chloroform. These are the different preventive measures one can adopt in order to prevent prevent this spreading of the COVID-19 pandemic. Now, I will come one by one. Now, how to use a mask? The mask guidelines for use of the mask. Perform by hand hygiene. Hands should be washed properly. Unhold the plates. Make sure that they are facing down. It should be placed over the nose, mouth, and chins. Fit flexible nose piece over nose bridge. Secure with the strings. Upper strings to be tied on the top of the head above the ears and the lower strings at the back of the neck. Ensure there is no gap on either side of the mask and adjust to fit. Do not let the mask hanging from your neck. Change the mask after every 6 hours or as soon as they become wet. Disposable masks are never to be reused and should be disposed of. While removing the mask, great care must be taken not to touch potentially infected outer surface of the mask. To remove the mask, first untie the string below and then the string above and handle the mask using upper string. Disposal of the used mask. Used mask should be considered as a potential infected medical waste. Discard the mask in a closed bin immediately after use. So these are the guidelines how a mask should be used, and I think there should be proper propaganda regarding this, because I have seen many masks are just lying by the side of the road, and it's the potential risk for spreading of the disease. So there should be proper awareness regarding this. Now, how to wear, this is a classical way how to wear a mask. The upper slide, the upper picture shows the right way and the lower picture shows the wrong way of wearing mask. There's some way of I have seen that some people are wearing masks, and there's the jokes in the WhatsApp group. This is going on. This is called mask. There's no mask. It's called my mask and mask and others. So this shouldn't be done. Now, other way what is being adopted in airport and others is thermal screening. But as I said, there are people who are healthy with just by measuring the temperature, it doesn't give any protection to the society. But it is still going on. In our college also, there's a fever clinic. We are screening the fever patient because all fever are not COVID, but there should be a high doubt regarding that the patient may suffer COVID. That should be kept in mind. So the steps of hand hygiene is a very old procedure. How to wash the hand. First, wash hands with water. Apply enough soap to cover all hand surface. Rub hands palm to palm. Rub back of each hand with the palm and other hand. Rub palm with fingers interlaced. Rub the back of the fingers opposing fingers. Rub each thumb close by opposing thumb. Rub tip of the fingers in opposite palm. Rub each wrist and opposing hand. Rinse hands with water. Use elbow to turn off the tap. Dry thoroughly with a single-use towel. Hand washing should take at least 15 to 30 seconds. This is very important because just by washing the hands, it can be prevented a lot. Now, the guidelines on disinfection of indoors areas, including offices. Office space should be cleaned every evening after office hours or early in the morning before rooms are occupied. Prior to cleaning, the worker should wear disposable rubber boots, gloves, heavy duty, and a triple layer mask. Start cleaning from cleaner areas and proceed to the dirtier areas. All indoor areas such as entrance lobbies, corridors, staircases, escalators, elevators, security guard booth, office rooms, meeting rooms, cafeteria should be mopped with a disinfectant with 1% sodium hypochlorite or phenolic disinfectant. High contact surfaces such as elevator button, handrails, handles, and call buttons, escalator, handrails, public counters, intercom system, equipment like telephone, printers, scanners, and other office machines should be cleaned twice daily by mopping with a linen absorbable cloth soaked by 1% sodium hypochlorite. Frequently touch areas like cable tops, chair handles, pens, diary, files, keyboards, mouse, mouse pad, tea, coffee, dispensing machines, etc. should be specially be cleaned. For metallic surfaces like door handles, security locks, key, etc., 70% alcohol can be used to wipe down the surface where the use of bleach is not suitable. Hand sanitizing stations should be installed in office premises, especially in the entry and near high contact surfaces. In the meeting conference office room, if someone is coughing without following the respiratory etiquette or the mask, the areas around his or her seat should be vacated and cleaned with 1% sodium hypochlorite. Carefully clean the equipment used in cleaning at the end of the cleaning process. Remove PPE, discard in a disposable PPE in yellow disposable bag and wash hands with soap and water. In addition, all employees should consider cleaning the work area in front of them with a disinfecting wipe prior to use and sit farther away from other if possible. Now, guidelines on disinfection of outdoor areas. Outdoor areas have less risk when indoor areas due to air current and exposure to sunlight. These include bus stops, railway platform, parks, road, etc. Cleaning and disinfection effort should be targeted to frequently touched contaminated surfaces. Guidelines on disinfection of public and common toilets, toilet pot commode, sodium hypochlorite 1% detergent, soap water, long handle angular brush, procedure inside of the toilet pot and commode, scrub with the recommended agent of the long handle angular brush, outside cleaned with the recommended agent, use a scrubber, lead and commode, nylon scrubber and soap water, nylon scrubber and soap powder detergent, wet and scrub with the soap powder and nylon, and the procedure is wet the scrub with soap powder and nylon scrubber inside and outside, wipe with 1% sodium hypochlorite. In this way, there are how the toilet floor can be cleaned. It should be cleaned with the soap powder, detergent, scrubbing brush, nylon broom, 1% sodium hypochlorite. Sink, soap water should be cleaned with the soap water, detergent and nylon scrubber, 1% sodium hypochlorite. Showers areas, taps and fittings should be cleaned with the warm water, detergent, powder, nylon scrubber, 1%, sodium hypochlorite, 70% or 70% alcohol. These are the things can be used for, agents can be used for this washing this different commode and floor, etc. Now there are two terminologies. One is the quarantine and the isolation. The isolation is the separation of the sick people with the contagious disease from people who are not sick. Now, and the quarantine separates and restricts the movement of the people who are exposed to a contagious disease to see if they become sick. These people may have been exposed to a disease or do not know it, or they may have the disease but do not show symptoms. So these two things have to be kept in mind for the prevention, because there are two important measures for to prevent the spreading of the disease into the community. In Kalyani, you know, there's a COVID hospital where we isolate, and there's a fever clinic in our college where we quarantine, where we keep the suspected patient. Now, who needs quarantine? Who have come with the patient or the person who has come within 6 feet contact with the COVID-19 active case for 15 minutes. Number 2, who provided care of the COVID-19 patient at home. Number 3, the physical contact with someone who is confirmed case. Number 4, exposed to sneeze, cough or respiratory droplets of an infected case, and who shared eating, drinking utensils with a confirmed case. As early as possible, so that they can the prevention, this spreading of the disease in the society can be prevented. Now, the prophylaxis prevention is always better than cure. And my earlier speaker, Professor Majumdar, was given a very good talk on this prophylactic basis. And you know, there are two varieties of prophylaxis. One is the chemoprophylaxis and one is the immunoprophylaxis. There's a very good talk on this immunoprophylaxis, and he has covered a little bit of the BCG vaccination, which is an immunoprophylaxis, but which is mainly made for the tuberculosis. But BCG has got a property to enhance the immunity in the same way. This chemoprophylaxis is also there, just like hydroxychloroquine somebody is giving. The other drugs are coming out from indigenous drugs. There are so many claimings from different houses that they enhance the immunity and it can protect you from the this COVID-19. Now, this is my personal view. There are other drugs like this. This is my personal view. It's not documented. Number one, the thalidomide, which was a drug used which was withdrawn from the market. Has got a potentially to enhance the immunity, and there are some literature perhaps coming out in different books that it can enhance this prophylaxis and can prevent this COVID-19. It is my personal view because self-protection is always important, and there's another drug which is not documented, but I personally feel as the pharmacologist has got maybe have some role in this prevention, and that is levamisole. Levamisole is used in so many conditions as an immunostimulator, but there are drawbacks. It may be probed. I think there are so many scientists in this conference, video conference workshop. They can give their view regarding this. Thank you very much for this listening. I think if there is any question, I will be able to answer which are pertinent to me. Now, one thing I forgot to mention, now interpretation, because there are a lot of confusion going on in the market, how to interpret the different readings of the RT-PCR and IgM and Ig estimation. The first is if the patient is PCR positive, IgM negative, and IgG is also negative, so the patient may be in the window period. Window period is the period when the patient got infected, but there is no manifestation of the disease on that period. We call it the window period. That means the patient is in window period. PCR positive, IgM negative, IgG negative indicate the patient is in window period. Now the PCR positive, IgM positive, and IgG negative is early stage of infection. PCR not done, IgM positive, IgG negative is early stage of infection. PCR test from oro and nasopharynx should be done as early as possible. Now PCR positive, IgM positive, and this IgG positive is an active infection. PCR not done, IgM positive, IgG positive is active phase of infection. PCR test should be done as early as possible. PCR negative, IgM, PCR negative, IgG positive, and IgM positive, and IgG negative is early stage of infection. PCR should be PCR result may be false negative. Now negative PCR, IgM negative and IgG positive is a post-infection recovered. PCR negative, IgM positive, and IgG positive is a post-infection, as I said. PCR negative, IgM positive, and IgG positive is a recovery stage infection, or PCR result may be false negative. Now, negative for the PCR, IgM negative and IgG negative is not infected or in early window period. These are the different interpretations what we can get. So this should be kept in mind before giving a stamp diagnosis to a patient of COVID-19. Thank you very much. Thank you very much, sir.
Okay, so thank you, sir, very much for your kind deliberation. So, and also you have finished before time, so a special thanks to you for that. So our next speaker of the session is Dr. Ujjwal Niyogi. Yeah, so it's my honor to introduce him because he is my school friend and presently associate professor at the Karolinska Institute, the institute which is known to distribute Nobel for physiology or medicine. He is also an adjunct professor, University of Missouri, Columbia, USA. To introduce his academic career in brief, so he did his MSc from the University of Bangalore in the year of 2007. Then he was in National Institute of Immunology for 2 years. After that, he joined a PhD at Karolinska Institute. He finished it in the year of 2013. Thereafter, he did his PDF postdoctoral studies from St. John's Research Institute, Bangalore. Thereafter, he joined Karolinska Institute as an assistant professor in the year of 2015. Now he is associate professor over there. He has fundings from several Swedish Research Council as well as from National Institute of Health, USA. His research interest revolves around the HIV immunology, metabolic profiles of viral diseases, but the present breakthrough of COVID-19, he is also interested to study that, and he is doing commendable job over there. So the renowned publications that he have is, some of them are, he publishes in Lancet Infectious Diseases, the Lancet HIV, then AIDS Scientific Research Reports, JBC Journal of Virology, and there are many more in the list. With this short introduction, I request Ujjwal to come up with his deliberation. Ujjwal, all is yours.
Yeah. Thank you, Dipanjan. I just need to check how to enter your screen.
In the extreme right corner downward, you see there is a present now tab.
Yes.
So before that, you need to open your PowerPoint presentation.
Yeah, it's open.
Okay, now in the present now tab, you see there is a second option, your entire screen and then a window. So just select a window.
Okay, yeah, now I can.
And then you highlight your presentation.
Okay. Can you see my full screen presentation?
Yeah, it's fine.
Yeah, it's fine.
Okay. Thank you, Dipanjan and Professor Ghosh, to invite me for this presentation. And thank you, Dipanjan, for the nice introduction. So I'm going to talk about therapeutic options in COVID-19 patient. I call it information and misinformation because lots of things are going, and I will try to convince you few of the things if you like that. So for today's talk, I'm going to keep very brief on general background and current situation worldwide. You might have heard lots about Sweden in newspaper, and several times those are very wrong information. So I'll briefly tell about the Swedish strategy, which is completely different than any other country in this world. And I will talk a little bit about the virus and the different therapeutic options. That is, we have direct acting antivirals and immune modulator, but my lab is mainly focusing on the host targeted therapy. So I will talk more about that. So as you already know that it is now spread, the COVID-19 is spread around 188 countries. And there is now some hotspots still going on in US, and then India is coming up. And on 30 July, the present current situation is like new cases are going up except few countries, and you can see the dark red colors in different places. So the Sweden is actually outliers in the whole response to epidemic, because when the epidemic started, Swedish government hand over the whole response to a scientific community. That's called the Public Health Sweden, and this is a nice statement. I think it's given by the Prime Minister Stefan Löfven in February, that the crisis will continue for a long time. It will be tough, but our society is strong. If everyone takes their responsibility together, we can overcome it. So it's the instruction from government is clear. It's your responsibility to save the society. And then they hand over the government never come into the picture much. So they hand over the whole response strategy to the Public Health Sweden, and Public Health Sweden is not only the public health, it also has economic expert and crisis management and response to any calamity and other thing. And this is the guy who I think now under fire from several countries, several prime ministers, president are talking about is Anders Tegnell, who is actually a SARS expert, and he in 2007 actually predicted that this kind of epidemic is going to come. So why Swedish is outlier? The first thing, there is no lockdown at all in the whole country since the beginning. So everything is open, but there's an instruction how to behave in the public places. There's no face mask at all. The schools are open. Only the closed thing is high schools and the university colleges, and they are not closed, they are distance mode. The reason behind that is because if the preschool, like children's school is closed, then they expected around 30,000 frontline workers like police, doctors, nurses have to be in home. So they decided, okay, we will take care of and the children, you take care of the society. And they only asked the younger group, like those who are more than 14 years of age, that school group until university to go distance mode because they can take care of themselves. So it's a famous, I think in last 3 years, he has mentioned several times, Donald Trump has mentioned several times about Swedish strategy and all are misinformed. There are debate going on in the scientific domain as well as in the article. I think BBC has covered around 20 articles on Sweden just to show that whether it is failure or success. But what we think, who are staying here, around 70 to 80% population believe the response is correct. And the most important part, so this guy is the Prime Minister, Stefan Löfven, is we believe the Public Health Sweden, and you can see there's a meet going on with all European Union. The only person who is not using mask is Stefan Löfven. So he even follows that one now, that okay, there's no mask is mandatory. But is that really true that there is no lockdown at all? It's actually not correct. So general advice from the Public Health Agency, and they have introduced the Infectious Disease Act. So it's a very clear instruction, stay home if you have ill or if you feel little bit sick also. Simple sore throat, you be in home, you will get sick leave or paid money from the government. Wash your hand frequently with soap and water, keep away from others, both indoors and outdoors. And from the beginning, it was 2 meters. And they put up big boards in all the bus stops and other places. Keep away from others, from bus, train, and they now make bus free of charge so that you don't have to enter to show your ticket to the bus drivers. Avoid funeral, baptism, parties, wedding, like all kind of religious and cultural event. Keep away from others at the sports venue, swimming pools, and other places. Do not travel during the rush hour and only travel if necessary. So with this, and one more important part, because that is a failure of the Swedish strategy, if you are 70 years of age or older, do not try to avoid the contact. So we have already shown about flatten the curve. All the countries are talking about but I am not sure anyone has flattened the curve yet. But what we can see, this is actually a real flatten the curve. This is a real-time data. And so it's highly increased in the early of March. That is because there are now 150,000 people came back from Italy from the sports weekend. And then the sudden increase, and then it's flattened, and then again increase due to capacity. And now it's come down. And you can see this is how the countries look. Some countries are flattened, some are going still up. Unfortunately, India is going a very high rate now. But Sweden is the only country, and another few, those who actually gone up and come down over the time. But there is a big failure from the Swedish strategy, that is to protect the elderly care home. So this is the nursing home or the old age homes, where mainly the people, those who are sick there, and they need a treatment. So more than 50% death in Sweden is from this nursing elderly care home. So if I go to, then the question comes, virologists like us and others, is it really a dangerous virus? Because if we compare 4 viruses like flu, normal flu, COVID, SARS, and MERS, I just want to point out one thing, is the annual infected. It's around 1 billion infected flu, where about in 6 months is 17 million, but SARS and the case fatality ratio, that the percentage of death, is 0.1%, it's around 3.9%. SARS is around 11% and MERS is around 34%. And this 3.9 also, you can consider less than 1% below age of 60. So rest of the thing is higher age. So that is the clear message is protect the old beloved in your household. And if you look into the different disease case fatality ratio, there are other diseases also we are now completely neglecting at this moment. So we don't know what will happen when the COVID is over. For example, like A, B, C, D, E, which are very low case fatality ratio now, and COVID is on D, that is 3.9. However, there is a dengue, which is now, I think, hitting in West Bengal and other parts of India, MERS, Ebola, bird flu. So they are much more dangerous than the COVID-19. And if we see the basic reproductive ratio, how we, the infectious disease epidemiology, like the ability of a virus to infect different, that's also not that high compared to measles, chickenpox, mumps, even Zika is high. So we're not sure the response and other thing. But yes, this is unknown disease. If we can protect our elderly people, I think the strategy can work. And this is the strategy of Sweden, try to protect the elderly people. If we go a little bit, I'll just touch upon different, the genome organization, because when I talk about direct acting antiviral, so it will help me to tell you about the target. So it has, the virus has 2 major ORF, open reading frame, that's called ORF 1A and 1B. And it has papain-like protease and 3CL protease, where there is a drug target, and when I'll tell about those. Then there's RNA-dependent RNA polymerase, a major drug target there. Then we have endonuclease, helicase, and spike protein, that is also a drug target. So if we see the course of the disease, because this is also important to understand the therapeutic response, or to know, because now in Sweden, we are able to come down to 1 to 2 deaths per day or 7 to 10 deaths per week. That is because initially we have a problem, because sometimes the patient look, they are healthy, they're discharged from the hospital, and what we observe is like as soon as they go home, maybe in 24 hours, they got a sudden heart attack. And then die. And then that time, the response time is less than 2 hours to bring them to hospital. And I think this is also...
আচ্ছা স্যার আমি আপনার ওই যে আপনি যে শিক্ষারত্নটা পাঠিয়েছিলেন না তার একটা ড্রাফট
Okay, so there's some noise from the background.
আপনি ড্রাফটটা অ্যাপ্রুভ করে দেন আজকে আমি শেয়ারে পাঠিয়ে দিই
Okay, so there are severe disease. There's a disease outcome in the onset. There's a viral exposure. Then one can get first symptoms up to 3 days post-infection, and then most of the people, 80% of people, don't observe any symptoms, while some may progress to survivors. The survivors, they will recover, and then non-survivors, they will.
অথবা আরেকটা জিনিস করতে পারেন দেখুন আপনার ড্রাফটটা যদি পছন্দ হয় চেঞ্জ
Is it possible to mute the
উপরটা চেঞ্জ করতে
who is talking in the background?
লিখেছি
ইন জেনারেল বলছেন তাই না?
হ্যাঁ নরমাল কাজে ইন জেনারেল সিআরএম এ যে 185 জনের নাম আছে পাঠিয়ে দিন ওখানে লেখা আছে যারা এলিজিবল এন্ড ইন্টারেস্টেড তারা অ্যাপ্লাই করতে পারবে আমি পাঠিয়েছি এই শিক্ষাটা হ্যাঁ এই যে এই ডট ডট লাইনের মাঝখানে একটু হচ্ছে আমার এইটুকু আমি অ্যাপ্রুভালটা ছিল এক্ষেত্রে এইটুকু চেঞ্জ করতে হবে
Okay, so
একদম 185 জনের কাছে চলে আর ওখানে লেখাই আছে যে প্রফেসর ছাড়া কেউ পাবে না এলিজিবল
ওখানে লেখা আছে
ব্র্যাকেটে হ্যাঁ প্রফেসর দেখবেন এটা খুলুন এটা খুললেই দেখতে হবে এটা যারা এলিজিবল এই 3T care.
Thank you. So the COVID-19 treatment options, if I go there, there are three options that we are currently looking. So direct acting antivirals and host modulators and host targeted therapy. So these all three are now either on emergency approval or we are going there. So this is the slide created by one of my master students, is nicely expressing where we can use different kind of drugs. So you can see this is in the beginning of the pandemic. So chloroquine, hydroxychloroquine, you have heard a lot of that. And then we have lopinavir, ritonavir, which is a drug that mainly developed for against the HIV and disulfiram. So that targets on the protease of the drug, and most of the drugs which work better, that's remdesivir, favipiravir, and ribavirin. That's also work on RDRP. So that's a replication complex, the polymerase domain, and then you can see this drug or repurposed drug are based on the theory or based on the its effect on SARS, MERS, or other human coronaviruses. So I will go a few of the drugs in detail. So this remdesivir, which is now mostly working and are in clinical practice, that is, so it goes and bind to the RNA-dependent RNA polymerase and stop the virus replication. In the beginning, very early, we used our computational power to try to see which kind of drug. We screened around 3 million compound, and we actually in the beginning we showed that it can go and bind to SARS-CoV-2 polymerase, and but we have some concern it may give rapid drug resistance to that. After that, there was some study that now come up that okay, yes, it's working in the cell culture assays. And the percentage is nice curve when they're working on Vero E6, and it's mainly worked after virus entered into the cell. So the percentage of inhibition is over. If you keep the drug for all the time, is good, but at the entry level, it's negative. It's not work, but post entry level, it works better. The first report came from China. Is not much encouraging. Remdesivir was not associated with statistically significant clinical benefit. And then there's a large study that done in US, and they also It can shorten the time to recovery in hospitalized patient, but no superior like effect on virus activation. So virus shedding, that viral load didn't go down. So it might have some other effect, not or it's not working on the virus as such. The second drug, which is very famous because even American president was also using, and he is ready to have a war with some countries, they don't provide them. So this is the hydroxychloroquine, and that is based on the data from France that it may work. So the mechanism of hydroxychloroquine, it can block the entry stage, early stage. It can block the MAPK signaling pathway, so then can stop the viral transcription. It can block interfere with the vacuole formation, so it can block the budding of the virus. And the lab data again show very nice. And when we compare to remdesivir, it has higher EC50 value, but the effect is also in the entry level. So this was a magic drug people has proposed, but it's actually derailed the whole situation because there is shortage for even malarial patient, and then hydroxychloroquine or chloroquine do not inhibit infection in human lung cells with SARS-CoV-2, and the paper which was submitted by the French group was retracted because there was some error. And then hydroxychloroquine used against SARS-CoV-2 infection in non-primate also give the negative result. So this actually delayed all the process of using other, and WHO was not sure. First time they stopped the trial and then again started, and it's now not recommended outside the clinical trial. The third drug is lopinavir ritonavir, so which is the anti-HIV drug. It has been shown that computer prediction, it has shown that it can bind to the protease. As these drugs are used, its safety profile is clear. The Chinese group and some of the Hong Kong group, they started with this drug. But again, there is no effect that was observed in hospitalized adult patient, because for the mild patient, they are the super spreader, but we have to save our people who are already hospitalized because they are getting, they are mostly dying. So then while doing this, some clinician has observed there's a huge cytokine storm. Lots of cytokine has gone up. So some doctors, they started, including our hospital and Karolinska Institute, like when there is no drug, so suddenly they start giving this tocilizumab, which blocks the IL-6 receptor and JAK-STAT pathway. And some people like Cuba got some effect on interferon delta, not beta here. So this can also be an option that will be more like supplementary. So it will not affect directly on the virus, but if our body take care of the virus, then we can give this kind of treatment to recover the cytokine storm. So this is the drug that is used, and it can block the IL-6, similarly to its sarilumab. But there's a study from, I think it's from Italy. They showed that it has effect on the reduced risk of invasive mechanical ventilation. And our study in Karolinska Institute has shown that it reduces the time of the ventilator stay, but it does not have any effect on the overall viral replication or mortality. So it's contradictory. So what we believe, and when I spoke to the doctors, they believe that timing when to give, because if these drugs given very early stage of the disease, it can be a detrimental effect on the patient, because it can also give some kind of shock. And so the timing is important, and so that's why it's a clear guideline or clinical decision is important. So now we'll come mostly to the host targeted therapy, which is more like new direction of infectious disease that's developed last 5-6 years that aim to interfere with the host molecule pathway or the network that are required for infection to contribute. Because when I come here in Sweden, when there is a viral disease, including flu, there is no treatment they used to give. They believe on natural healing. Like when we have fever, just they said, okay, 5 days you stay home. If still the fever don't go, then you come, then we'll treat for bacterial infection, if that is positive, or else they generally recommend to stay home. So from there, this idea came, okay, can we use this strategy, host targeted therapy? And then I started looking for that, and I found this has been actually, people are trying on TB because of the drug resistance. So what is that? What is the mechanism of host-targeted therapy? This is mainly modulate the inflammatory pathway and proliferatory mediator to dampen the inflammation and the inflammation-induced pathology. You might have heard about the bat, seahorse bat. They are the carrier of the several coronavirus, but they never show symptoms. So one of the important thing for them is somehow they are able to keep the inflammation level low. So they dampen the inflammation so that they can carry those viruses. And then to enhance the host immune response to efficacy and strengthen the immune and memory response, and enhance host virucidal mechanism. So how we work in the lab. So we already work, our major focus is on HIV, but we already work with Crimea Congo hemorrhagic fever, and we are also working with Ebola. And then suddenly the SARS-2 came and we just applied. So this is the fastest method where we applied the whole host targeted therapy mechanism, and we get the data in actually a week. So how we do, we try to, if we try to work on a cohort of patient who had different disease status, and if that is not available in case of SARS, we first started with the in vitro infection model. And then we go for multi-omic system biology. So in our lab, we are experienced mainly on transcriptomics, metabolomics, and proteomics. And then we try to find out, okay, where the virus is targeting, what kind of gene expression is happening. And then when you get the this target, luckily in our case in SARS-CoV-2, it is the pathway that we identify, which we are working on for HIV functional cure. So then we go for mechanistic study to give different pathway modulator to see whether it can control the viral replication or what. So, and in the lab, we are mainly working with HIV functional cure using a patient group called elite controller who control the viral replication by themselves without any therapy. And the respiratory virus we just started because I was working with viral hemorrhagic fever, CCHF, Ebola, dengue, and Zika. And then respiratory viruses like SARS, SARS-CoV-2, H1N1, and RSV. So this is the broad, but the overall approach is same. So we have everything automated. Pipelines, the lab experiment mostly is computer, like robotics. So we just extract RNA, put it into the robot, and to do the next of the process for transcriptomics, same for proteomics and metabolomics. So when this virus first came, we first identified with an isolate from a Swedish patient who came from Wuhan. And we tried different cell line because it was the initial notion that it will infect the lung cell. So the 16 HBr lung cell. But unfortunately, this is puzzling that it do not infect maximum lung cells. So as of now, we have tried around 5 lung cells. Only there is a moderate infection in the Kalu3, which is a lung epithelial cell line. So we didn't find anything, and then we found HU7, which is a kidney cell line. And then we started doing to understand the cellular, how the virus affect. So we infect the cells with the virus, and then we use transcriptomics, proteomics, and proteotranscriptomics, and we also identify the virus and see the dynamics of the virus, how it looks, and then we choose 72 hours as an overall time course experiment. To our interest, what we have observed when we use sophisticated. I'm not going details of the bioinformatics analysis, which is the HIF1 and mTOR signaling pathway. And when we are doing the analysis, we also identified one paper came out, which also identify HIF1 signaling. This HIF1 mTOR signaling is the central pathway of life, because we were working on that because of we want to identify the functional cue to modulate that pathway. And when you use more sophisticated analysis, and we identified that, so this analysis is called centrality analysis. So what we observe, if a community of proteins or genes or metabolite in the middle, so they regulate the others one. So we found there several proteins are from the mTOR signaling pathway. And then we went further to see whether virus can affect that. And we first characterized, yes, virus can affect. You can see dose-dependent analysis, and it's going up, particularly for HIF1, it's going down, which is strange, and then going up afterwards. We also notice when HIF, the hypoxia factor, upregulate, there are several genes can upregulate, and we can see several genes are upregulated in the figure E. And then we use RNA scope to see, to visualize whether it is fine. And you can see when at 48 hours, when SARS-CoV-2 infected, there are less number of F1 mRNA. Based on that, we targeted this pathway with 8 different drugs to target different level of this, from upstream PI3K to downstream HIF1 alpha. And what we have observed, that it has effect in different proteins. And to our surprise, we have observed that MK2206, which is actually target the AKT, the first molecule here, MK2206, first molecule, and it can actually stop the viral replication. And now there is another paper in the bioarchive they have shown is, yes, it's work. But the SAHA, which affect on multiple level, because if you see the Western blot, it affect HIF1, it affect S6K, it's upregulate S6K, and then it affect AKT. It increases the viral replication. So touching this pathway need to be very careful. So during that time, we saw a paper that open question, is it rational to inhibit the mTOR pathway, COVID therapy, and they proposed it. But still, we are not sure that, but already the clinical trial started in June. So we'll come to know whether it has any effect on reducing the severity in COVID-19 patient. So if we go for the conclusions, so no antiviral are effective in reducing the viral load in patient. This drug, remdesivir, hydroxychloroquine, lopinavir, can work in vitro, but in patient, no such info there. Then remdesivir does not have any clinical benefit in severe patient, but it can shorten the time to recovery. Then severe patient needs special attention to reduce cytokine storm, because now in India there is one Biocon drug is introduced to reduce the cytokine storm, but I'm not sure how many hospital can do this measurement of cytokine immediately when to deep, because if you give that drug in the early stage, it could be a detrimental for the patient. mTOR pathway can be attractive strategy, but in combination with antivirals. And studies should go beyond the computer prediction, because all these drug repurpose and all those data is based on the computer prediction, whether it's binding to the drugs or not. It should go beyond that first to identify the virus, because we have technology available that one can screen. I think the paper is now online from one of the German group, that you can screen 30,000 people against a virus if the virus is available in like 5 to 6 days. So rather than computer prediction, better to go to the virus biology. But there is a hope, but people are not talking a lot about this. This is from Hong Kong. So they give a triple therapy, and I may be biased for triple therapy because I'm working on HIV field, and triple therapy is the only option there. So triple combination therapy is like combination of interferon beta, 1 beta, lopinavir, ritonavir, which is an anti-HIV drug, and ribavirin. And then it actually reduced the viral load. It also reduced the symptoms and the shortening duration of the viral shedding, as well as hospital stay. So there is a trial going on in Karolinska Institute. Hopefully we'll come to know soon. So the take-home message, as of now, no drug can stop the virus as such in patient. No vaccine, hopefully, and the data is coming up. So hopefully we have the vaccine soon, but it will take at least a year from now. So what we can do now, wash your hand frequently, cough and sneeze into elbow, avoid touching eyes and nose, and stay home if you feel unwell. So that's the Swedish strategy is there. So finally, I'd like to thank my team. It's a PhD student, Anup, and Flora, who are bioinformatician, and Mike, Shohom, and Shub, rest of the thing, they're in the lab. And I should be proud of them because we started isolating the virus in March end, and now we have our first paper online in like 2-3 months, and which is without any knowledge, any experience how to work. And this also to work in P3 because we don't know the pathogen, how it is. And the biosafety level 3 is recommended in Sweden to work with SARS. And it cannot be possible without the large interdisciplinary team. So I should also thank to the proteomics, because proteomics study was done in 2 days. So 3 days for infection and 2 days to do LCMS based analysis. And then the transcriptomics and other things, we have all national infrastructure to do. Now we are continuing. We want to see how this is there in the patient. So we are now continuing clinical study. I have 3 cohort. One is in China. So I want to see, our interest is metabolic syndrome in HIV. So, and what we know from the doctors that this patient is severely metabolic distress is there. So we want to see long-term effect. So we have a cohort in Italy, which is one of the hardest state, where we have collected at the time of hospitalization, at the time of discharge, and we'll collect after 6 months. In China, so this is again at the time of infection, hospitalization, and 1 year after that. And our like robust clinical study that we are collecting every month sample from South Hospital, Sweden. I have some also collaboration with Dr. Jayashree Das Sharma and ISAR Kolkata. We have tested their neem bark extract, which actually has antiviral effect. And I share the data with her, and hopefully she will continue doing that one. And we will go for animal model because we identify already one target for thing, which is with University of Nebraska. Thank you for your attention.
Thank you, Ujjwal, for your nice deliberation. Now the session is open for question and answering. So few participants. So Ujjwal, YouTube streaming is also going on with this. So they are having some questions which I want to share with you. So yeah. Aparna Banerjee is asking, can COVID-19 spread through water? Can contaminated drinking water cause infection?
As of now, no. Because there is some study reported, I think last week, that simple water can kill the virus and hot water can kill the virus. But there is viral residues, like PCR positive viral residues can be identified in the fecal material, like in the sewage treatment. So I don't think it can spread by water.
So if the virus is kept into the water, so how long it survives? Do you have any like idea?
It's the study says it's actually 3 hours, less than 3 hours it survives. But that study has a different, because they boil the water. So
okay.
It's like simple warm water is enough to actually kill the virus. And even simple UV is enough to kill the virus in 3 minutes. So when we take out the virus thing from the lab, from the biosafety level 3 lab, we go for 96 degrees centigrade for 3 minutes or UV treatment for 3 minutes. That's all.
So another question is, what is the role of natural killer cells, which is known to have essential importance in case of viral infection?
Sorry, I am not immunologist, so I can't respond you correctly, but there is some paper came out, it has some role, but which role is not, because all the paper now coming up is, actually there's a good study going on in our institute. We call it the Immune Atlas. So they are actually doing typing of different cell populations. So it's T cell biology, B cell biology, and NK cell biology. They came together and then they are now typing. So what is that effect is still like all a theoretical effect, not that much information. But
Right.
certainly, if you go by theory, there should be an effect.
Right. So another question is, SARS-CoV-2 is highly mutated nowadays. So
No,
for
it's
drug
wrong
treatment or
information.
vaccine-
It's wrong information. It's not highly mutative. It's a selection of one particular strain called D614G, and it's like 10 mutation in last 6 months. It's not that highly mutated than H1N1 and others. So it's a selection, and then it's just a transformation. It's also not that. So if the epidemic has started with D64G, it will spread more, and that's the natural phenomenon. But I don't think it's mutating to a dangerous strain and other thing. No, certainly not.
So someone is asking only about the immune therapy. So how effective it will be, like interleukins therapy?
So interleukin therapy alone, I don't think, because doctors are still not sure what to treat. It's not a simple lung disease, which that is the reason, like initially it's thought a lung disease and then people started treating. And what happens mainly now is like you discharge the patient thinking that virus is not there. Suddenly within 2 days or 3 days, this expired. And I think this is now happening in India also. There's some report that they discharge the patient and first 15 deaths actually in Sweden is like that. They thought patient recovered, they left them, they go home and suddenly stroke and died. So it's also a vascular, so it's affecting the blood. It's forming the blood clot, thrombosis. So that can, so it depends, and it's actually need very clear guideline for the treating doctor, because all are not experienced doctor. So when to treat, to what? But interleukin therapy will certainly, there is trial going on because IL-6 blocker, it worked in some country, it didn't work. It didn't work does not mean it do not work. Maybe they have administered at wrong time. It worked in Karolinska, this IL-6 blocker, it worked because they just, one doctor, as junior doctor, seeing the cytokine storm, he gave the medicines and they see the patient come out of ventilation after 2 days. So they said, okay, let's give. So it's everything is timing, when to give and how to give.
And another question is, sir, if remdesivir work by attaching to nucleotide of RNA and thus stop viral infection, why not it harms human RNA?
Its effect is go and bind to the RNA-dependent RNA polymerase of the virus. So, like it's a polymerase that going and binding. So not the like human RNA, it will not go bind because there will be certainly a side effect, but its off-target effect is not that much.
Right. Sir, fur of animal can transfer COVID-19 virus?
It's now identified in cat, but transferring, don't know as of now. Maybe it can transfer because some places is dog, the pet, and they thought that it can transfer.
Right. So I do have one question. So Sweden is the model which follows herd immunity, right? Although
no,
you showed the things how it is. Yeah.
It's.
But
so hard. Yeah. Continue.
In context of India, So where the whole population is like 130 crores or 140 crores, whatever it is. So do you think it is possible here, or only the country like advanced country like Sweden it is possible? So your comments.
No, I will not advocate for herd immunity at this moment, because it could have easily achieved if the lockdown was properly implemented or properly placed. It was like more like a midnight drama. It happened within 4 hours. It's lockdown, and no country did like that. They actually give time people to mobilize in different places to settle, and then so it's not possible now. So only option now is cluster lockdown. So if you identify more people in a cluster and then lock down that area for 1 to 2 weeks, it's too late for herd immunity concept. And actually Sweden is not herd immunity. It's more like it's the society thing, because I can see in the bus there is not much people. So and people are keeping distance among themselves in office and lab also, like those who can go work from home, they are working from home. And if you are little sick, like if you even have sore throat, government is paying you the sick leave benefit. So these are a small step. I don't think it's applicable to India. Now India only option is search more people, do more tests, and have more like advertisement. If you are sick, go for testing and then do a cluster lockdown. That is the only option now.
Yeah. So Akash Bishal sir, is there any other questions from your?
One question was asked from YouTuber, I guess, that it is something related to diagnostics. So in some cases, the patient was diagnosed to be positive, but after a few days, he's diagnosed to be negative. But again, that patient is showing symptoms of COVID-19. So can you please enlighten why these things happens?
Yeah. So first of all, this diagnostic thing is very important. How you are collecting the sample, how you are transporting the samples, and how the kit is sensitive for detecting the virus. So what it can be possible, the reinfection is still not that. It can be some residual viral thing that can come up later stage. Viral RNA copies know the active virus. But it's true that if the person is recovered from COVID, there are around 200 cold virus, and that is a season here. So it can be other virus also, because in the beginning, what I was a bit confused, people are talking about symptoms. What is the symptoms of COVID-19? Cough, cold, sore throat, fever. So all four are there for any general cold symptoms. So how you differentiate between? So that could be other virus. There is also possibility for false negative. So if you look in the data in the this world meter or something, Sweden is the only country who are not giving any PCR negative status at this discharge, because there is no. So what we follow, 14 days from the PCR detection, whatever it is. After 14 days, if it is there, the virus is non-infectious. Because the study is done in UK on healthcare professional, and they find out that after 7 days, like 25% patient only has the active virus replication, and after 14 days, it's come down to 1 to 2%. So it could be the kit that is used, how the samples are collected, and those kind of like more diagnostic, not scientific or clinical.
So is there any other? Okay, if not, we would like to pay thanks to Ujjwal once more for your time. I know it's like early morning over there at around like 6 or something like that, 6 a.m. or something like that. So it's really tough for you to join us. So once again, thank you very much. So we are almost at the end of our program. So all the deliberations by our eminent speakers are over. So now I would like to ask our coordinator DST Pars, madam, if you are here present. So can you please speak a few words?
Very good afternoon to all of you. Today's set of lectures was as exciting as the previous 2 days. And I think to give an overview, we began with a lecture by Professor Soumitra Das, the director of NIBMG, and he spoke at length about RNA viruses with special references to challenges by COVID-19. So I think it was a very good start. And then our Honorable Vice Chancellor, Professor Shankar Kumar Ghosh, he spoke about social distancing, the do's and don'ts that we need to know and to follow until that there is a proper vaccine available. Then in the next day's session, we had Professor Kuhupal talking about the management of the disease, the different testing protocols. Then Professor Nita Bhattacharya, he spoke about the molecular biology of the virus. Next, Dr. Patru, he spoke about the nanotherapeutic approach, nanotechnological approaches in providing protection and prevention and also for detection of this virus. Today we had Professor Dr. Keshav Mukhopadhyay talking on the safety measures and the precautions to be taken during this period. Then we had, before that, we had a talk by Professor Boshanjit Mozumdar. He talked about the causes of the fatality, the pathways, what are the proteins that can be targeted, and why does the severity of the different, why does it vary, the severity of the disease vary between different patients, the strategies for developing different vaccines. And then we had Dr. Nyogi talking about the therapeutic options with particular, and he shared with us some of his findings in this area. So I think we had an excellent set of lectures where each single lecture reinforced the basic concepts about the biology of the SARS-2 COVID virus, the COVID-19, which is responsible for the COVID-19 disease. It has helped to enhance our knowledge and it has given up new informations. And I think I'm personally enriched after listening to all these different lectures. Then we had also a presentation from Thermo Fisher who showed us about who explained about the protocol involved, the technique involved in the RT-PCR, the RNA isolation and the RT-PCR technique. And I think it was a rare opportunity that we could see the real testing in the hospital settings. So overall, I think this was a very successful event. And for this, in the very beginning, I would like to thank our Honorable Vice Chancellor, sir. He has been the inspiration behind this workshop, and his active advice and help in every stage has made this made it possible for us to organize this workshop in a very short time. I would like to thank Professor Mukhopadhyay of JNM Hospital for collaborating with our university, and that's why we could also show the testing procedure in the hospital, and for his nice talk. I would also like to thank our three scientists at the DST Center. They have been taking endless pain not only for this workshop, but for each and every workshop that has been organized so far by the DST PERS under the DST PERS program. So I would like to, nothing would have been possible without their active cooperation. I would also like to thank our CRM Center at University of Kolani for providing all the infrastructure. My special thanks to all the speakers because with their enriching talk, I think this has gone a long way to the success of this workshop. I would like to thank all the participants. That it is because of them and because of their the nice questions they put forward to the speakers that made it much more interactive. So if there are any other queries by any participant, we would like to take it. And before we conclude, I think I will ask the STPL scientists, Dr. Dhar, Dr. Guha, and Mr. Akash Bhattacharya, if they could we have, I think, a very short video showing all the activities of the DST PERS program in our university in the last 1.5 years. So this will enable all the participants to see what are the facilities we have here because it is open to all. So with this, I would like to give it over to them.
Thank you, madam. Akash, can you play the video?
Yes, I will play the video.
So thank you very much for watching the video. So just one announcement, all the feedbacks form to the participants will be sent by email very shortly. So we will short the feedback forms to the participant very soon. So now I would like to ask our Honorable Vice Chancellor, sir, to say a few concluding words. Sir? So am I audible?
Write to him in the chat if he's perhaps he didn't hear you.
Akash, can you please type for sir? I think my mouse is not working properly. Sir, we are waiting for your concluding remarks. Yes, sir. Sir, we are waiting for your concluding remarks.
Sorry, I'm in the meeting right now in the examination session. When Rita Di called me, just
Okay, sir. We can understand about your busy schedule, sir.
So the examination meeting is going on, all of that there. So it's a just finishing touch or end?
Yes, sir. Yes, sir.
Okay.
Your remarks
Okay.
will be the concluding remarks.
Okay. So firstly, thanks the organizer, particularly the three scientists and the uh, uh, two COVID testing scientists. They are really actively engaged to organize this type of courses, virtual. I think this is the virtual workshop. And behind this entire workshop, the coordinator, Professor Rita Ghosh, is one of the milestone to give this kind of test to the participant, as well as the Kuhu madam and the principal JNM. They are arranging in such a way. So always we want to tell the interdisciplinary approach. This is a one kind of interdisciplinary approach where the basic, applied, and the testing part is organized in such a way, and wonderful discussion from the morning today and last 3 days, and really there are 8 or 9 different speakers are there, and lots of chat box I have found. They are really appreciate this program. I hope this will be very nice program. And moreover, from our DST parts, the Pratishtha Pandey, she is also announced that we are the one of the excellence DST parts unit out of 5 in the country. That is also a big remark from her side. And what I want to say that this is the just beginning. During the pandemic COVID period, this is the just beginning, and you just frame out another program, so that it makes the more vibration in the field of application, research, and pandemic situation, what science can do in the under the aegis of DST parts. Also, you can feel, or we can start some kind of science promotion with the National Academy of Science in collaboration with them. So we can plan on that area, and another more virtual workshop can be addressed, like instrumentation, genomic diversity, whatever you past experience are there. Now you can start this kind of virtual experiences, so that student should gain. Yesterday, one of student phoned me basically about the excellent deliberation of this workshop. Excellent derivation, and he used to take some pages and write down like a note, it's very amazing. And hopefully this kind of student can we can give a more excitement through this our program. I believe that new normal life will come, and we will be sustained, and we will be enjoy every time through this virtual mode. Once again, I really appreciate Professor Rita Ghosh to get in touch very nicely. She has made a mediator to scientists, to the organizer, to the authority, and give a very good successful program. I really congratulate her to give the patients and the right directions so that the university flagship can be achieved in a big way. That's the only part. And all the participants and all the speakers, in the morning, the Barshanjit is my senior. We worked in the Cleveland Clinic, and I know he's a very prominent scientist over there, and our university alumni from the Department of Biochemistry, Sujan. Sujan is his postdoc when I was in US. He was there as a postdoc in Cleveland State, Ohio. So overall, that's fine. Only the discussion part with the Barshanjit, there are lots of questions has come up. Hopefully, if you arrange in near future one discussion on this COVID and their pandemic part, we can arrange for 3 or 4 scientists at a field, and also we can invite all this part, and that make a sense. So what your next, you have to get the feedback form. You should just start some kind of evaluation process, because you have sent the more than 100 link, but right now there are only 62. And there are lots of YouTube viewer. First day I have seen that 2.3 K viewers are there. So those who are viewing all the part, you can make a one evaluation, some kind of evaluation process, and if you decide, we can give some kind of prize to them, those who are given the excellent in that way. So try to make a 5th September is our Teachers' Day, whether we can able to get a mega event with these digestive parts by covering some of the good people and give some kind of feedback and some kind of notions to our youngster particularly. With these few words, I once again thanks to everybody, to viewers, to all the part. This is a concluding remark from my side. Thank